Good.
All right, I'll start it now.
Perfect, thank you.
Then it'll work.
And then it'll start working. So if you wanted to advance slides, you have to click twice because the first time it's just selecting it, and the second time it's actually clicking. So same with the keys. You can click it, now you're selecting. These arrow keys are clicking.
Jerry,
All
should
right.
I bring Randy Oliver up?
Yep.
Okay.
This area or here where there's not a lot. Jerry, can you hear me?
Yep.
Okay, I don't know why. Boy, I could barely hear you guys. Just now got some volume.
Yeah, well, you're coming through fine now.
Yeah.
Okay. I don't know why I can barely hear you guys.
Have Frank check your audio under your general settings. You may have got the volume turned down.
I've got everything full blast right now.
Yeah, it should be fine.
So Jerry and Chris, I'm assuming, or whoever's going to do an intro, you'll remind
I
people
will.
use the Q&A and
Yep. I'm using cheat sheets tonight for a change. So
awesome.
you may see me reading some things, but I want to move things along tonight. So
Sounds
Well,
great.
that's for the chat. Eh? I can see. Oh, no, we can see this.
So Albert, what'll happen here is if Medhat comes on, I'm going to ask him to moderate the questions when we get to that. But ask you to try to keep you've got the first hour. Like you to try to keep your presentation down to about 45 minutes so we have some time for questions. The people asking questions will use the Q&A at the bottom to type their questions. And then whether it's Medhat or Etienne, I wasn't sure, Medhat often shows up. If he is, I ask him if he'll moderate. If not, Etienne from the Yukon territories will be, we'll go, when we go to the question and answer, he will read off of the Q&A kind of a summary questions from what's coming in and toss them to you and Ray. So, so
Okay.
you don't have to worry about watching Q&A and chat. One of them will.
Sure. Good. I don't have to worry.
Yeah, you just talk
Yeah.
and for 45 minutes or so, I'll yell at you and you try to wrap it up so we can get some questions in. And then we really want to get done by the end of the hour so that we can have a brief break. Rich Morris from Broodminders, the sponsor, will chat just for a couple minutes, and then we need to get to our general meeting, our award ceremony, and our election of new officers. So it's going to be a busy meeting, a busy evening.
Yeah.
All right, I got to go find
Right.
Medhat.
Yeah.
I see something from Medhat right now, so he's out there somewhere. And
Yeah,
Medhat, we're working on promoting you.
he's coming in.
There he is. All right, Medhat, turn on your microphone and chat with me.
Phew.
Your microphone's still off, Medhat. There he is. Now,
Yeah.
there. All right, Medhat.
Yes, sir. How are you?
I have two requests for you.
Yes.
Number one, when Albert gets done with his talk, and Ray, who's partnering with him on the Saskatraz Hive breeding program, when Albert finishes at about 45 minutes, will you be the moderator looking at the Q&A to ask some questions from the audience of Albert?
Absolutely.
All right, that's my first favor, and I assumed you'd be up for the game here and so on. Secondly, I asked that you be sure that I know it may be getting late for you, us old timers. I took a nap today, so I cheated, but please stay for the general meeting. I have something that will be of interest to you.
Okay, I'll hang around.
All
Even
right.
though I am working since 5 o'clock in the morning to finish a proposal, so.
Yeah, I understand, but it's worth your while to hang around. I'll put it right up what I'm talking about up at the very beginning of the general meeting. So hang in there. Then you can leave. Well, you'll know what I'm talking about when it happens.
As
But
long as you buy me a beer next time, so.
I will.
I'm
I'll buy
happy.
you a whole pitcher.
Hi, Bed Hat.
Here's to you,
Hi there.
Matt.
Yeah.
My coffee.
All right. It looks like we're right at 7 o'clock. How are we doing? 84 people in. We'll probably get a few more, but I'm going to try to maximize the time because we got a long evening here. So welcome to everyone that is tuned in for tonight's mini conference, which will be followed by the annual meeting of the membership of the Western Apicultural Society. Our sponsor tonight is Rich Morris from Broodminder, a high sensor system. And he will be talking a little bit about that between Albert's talk and the meeting for the Western Apiculture Society. As always, we will start with our speaker, followed by a Q&A session, or question and answer. Dr. Medhat Nasser will be moderating the Q&A. We remind everyone that with this webinar program, which allows us to reach a large crowd, that to conserve bandwidth, we are going to, you, the attendees, do not have live microphones or video. We ask the panelists who are on that when the speaker is talking, that if they would blank out their video and turn off their audio, it'll save on bandwidth and be a little less distraction. But you can do that. Wait. I'm really glad to see two of the long-term members and supporters of WAS already online. Dr. Eric Musson, who is the founder of WAS, and obviously Medhat, who's been with us for a long, long time. So on the bottom where it says Q&A and chat, use chat for I've got a problem. Can you do this? Can you do that? Howdy to whoever you want to shout out to. Use the question and answer specifically for the questions that Medhat will ask of Albert and when he's done. Now, our speaker tonight is, and at the top of the hour, after Rich Morris gives a little briefing on Broodminder, we will move to the annual meeting of the Western Apiculture Society. We're going to have an award ceremony. We're going to have an election. We're going to have a general brief overview of what's happened in the last year. For all of you on these mini conferences, if you're not a member of Western Apiculture Society, we're going to offer a 2-for-1 deal for you this year on dues. So, and you're very welcome to attend and participate in our in our general meeting. The only thing we ask you, you're on the honor system, don't vote in the elections. Only WAS members that are paid should vote in the elections. And I see some things flipping on here. That Randy Oliver's joining us, and Rich Morris said he's here. So I think everybody's here. Albert has also joined here with us. His partner has also joined us, Ray Oliveras from Oliveras Honeybees in Orland, California. So I'm going to turn this over. So Albert Robertson is with the Meadow Ridge Enterprise Group. He's in Saskatoon, Saskatchewan. And for the last few years, he's been doing a really remarkable queen breeding program that merges state-of-the-art technologies and methods, analytics and so on, experimentation and so on, in his quest for a better queen. So Albert, I'm going to turn it over to you. I'm going to blank my screen out and my audio. And the next time you hear from me, I'll be notifying you that your time's running out. We need to take some questions. All right? Go ahead, Albert.
Yeah, thank you, Jerry. Thanks for inviting us, and I'll rock and roll here. So about 2004, we started the Saskatraz breeding program with the objective of producing productive, gentle honeybees with tolerance to mites and brood diseases. And the objective, of course, remains the same. I think everybody would be agree with me that right now our current honeybee health issues are Varroa mainly, and the viruses that it transmits, and interference by miticides, pesticides, and nutrition. So all of these can combine to give us some problems with the honeybee health issues. So what I'm going to do tonight, I'm going to try and briefly review our breeding program. As far as biomarker work, I'm going to concentrate on genome arrays, a little bit on RNA-seq or microarrays. And we've started in the last few years screening Saskatraz families for virus susceptibility. And then I have a section on our hybrid project with Oliver honeybees in Orland. And the last section, if I have time, we'll look at combined miticide treatment experiments with selected and unselected stock. And if you you can visit our website, we've got our publications listed on there if you wish to read them. So the program began in 2004, and our primary selection criteria was honey production. That's because in our neck of the woods, we make our living from honey, wintering ability, spring population growth, and Varroa resistance and suppression, resistance to brood diseases, chalk brood, AFB, EFB, etc., which seems to go along with survival breeding. Viruses and nosema susceptibility we're working on, although I've dropped the nosema business. The breeding methods I'm going to talk about are mainly natural selection. We use outcrossing and backcrossing to maintain the lines. And the main theme is recurrent selection. Progeny analysis is becoming more important to us, and this is all centered around close population mating. So this was the original Saskatraz yard site in 2006. We started it in 4. That's the original crew there. And about 2010 or so, this yard was flooded, so we had to move the bees out, but we've set up another few locations. Nothing ever gets out of there except what we graft from to multiply the stock. These are some of the people involved. I'm with Philip Griebel and Wayne Connor at Vito. We've worked together for a number of years. We've had several graduate students. Sanjay Yang there was involved in to the right, was involved in the microarray analysis. And I have 2 fellows that help me with queen rearing, Edmundo and Antonio Manoz from Mexico. Their father was the president of the Queen Breeders Association in Mexico. So this is Saskatraz breeding program logistics. It looks complicated, but really it's not. The first part on the left is what we started with. We looked at, we'd been breeding at Meadow Ridge for honey production and wintering ability and chalk brood resistance, except for many years, since 1992, using closed population breeding. However, when the Varroa come along, we had no resistance here at all, or tracheomite, we had no resistance to that. So we brought in Russian selections with the SBA and the Ontario OBA. At that time, I think MedHat was around in Ontario, and we brought those in. The German semen we brought from Ralph Buchler's program, and several Russian selections from the USDA. And we collected stock from Saskatchewan and Manitoba, people that had been raising their own stock for a number of years. So selected for wintering ability and also for various brood diseases and honey production. In 2012, we got some BSH semen from Bob Dank at the USDA and inseminated some of our colonies in California, and mainly some of our best breeders that we used for that. So everything, we picked the best, put them into a natural selection apiary that I just showed you. No treatments were administered, and we outcrossed the survivors. And we also select from about 50 acres for honey production, wintering, and spring population growth. So we back cross, of course, the best colonies back to the Saskatraz apiary to try and increase survival, and keep cycling this procedure. And we've done that for a long time. We first started stock distribution in 2006 in Saskatchewan, and now through the hybrid project we're reaching quite a bit farther to various places in North America. So just to flesh out the close population mating procedures, we have natural selection yards. At one time I had up to 5. Right now we're just running a few. We do economic trait selection for honey production and winter survival from about 50 apiaries with 1,500 to 1,800 colonies. We assembled those in 3 yards that are within close proximity, so we have good drone congregation areas. And we have actually started to do a lot more progeny analysis lately. We try to get daughters from up to 30 daughters from our best breeders. And we do selection within that population to pick the best producers, the most tolerant to Varroa, and so on. So that's been useful, and I'll show you some examples of that later. So right now I'm going to go into sort of the detailed work that we do each year. And this is natural selection for Varroa tolerance. And you'll see on the y-axis we have the phoretic mite levels. And on the x-axis we have the colony selections that are in the apiary and the year that they were established. So in this particular case, 2014 was the oldest in 2018. So this colony has survived up to this point. And you'll notice on the, also along the x-axis, we have for each colony, the mite levels in the spring are in the green, and the mite levels in the fall in the blue, and the mean mite count over the year. So you see this one has, this SY26 has very good mite levels, very low even after 4 years. And we also have a 2016 colony that has better honey production, but still the S14 is pretty good production as well. So we would multiply these and we would cross it to our close population yards and back to the natural selection yard. This is a 2019 from the same colony. Some of them have died, but just to show you our 2014 selection is still alive. However, the mite population is starting to increase. In the spring, she was showing higher mites than she had for a while. And then in the fall, quite a bit higher. So she died in the spring of 2020, which was quite upsetting to us. But anyway, here's another example of a 2018 colony that showed excellent honey production. So she would be selected and her progeny multiplied. Selection for honey production, we do annually as well. And this graph shows honey production in pounds for 2019, 2020, and 2021. Now, this particular selection was made in 2019. However, it's the result of a cross between a progeny, 2 progeny yards, where we crossed the SL25 was showed good Varroa tolerance to our best honey producers. So this ended up giving us quite a good kick in honey production. This S28 is one of my favorites. She's been around, this family has been around this strain since 2006. And one of the interesting things, you'll find in this colony often that she has a daughter. The mother and the daughter will be cohabiting and both laying, and that's why I think their longevity is so good. She started to fail a bit in 2020 because of that. She's coming back up again to 250, and she done very well in 2019. We now have the full genomic sequence of this strain. And this is, I'm going to go through some of the work we do on progeny analysis. This particular slide is quite old, but it's a good example of how we do it. The percent Varroa infestation is on the y-axis there, and the progeny. I have 8 daughters here from our S13 breeder, and we measure the mite levels in the fall, in November, and then we remeasure them again in April. And this is a good way to select colonies that when you're going through a broodless period, that through grooming activity and aggressiveness towards mites, there's few mites in the spring in these colonies. So we would select these for further multiplication. Nosema, we've also done that, but I've had a hard time keeping people able to do that. It's not the best job in the world. The number of spores on the y-axis there, and we have 8 daughters here that we looked at from December to February, and we would of course not pick the S88-3, but we pick SA88-5. I've discontinued. I've given up on Nosema selection. We just don't treat for it, and the ones that are susceptible are eliminated from the population. We did a lot of mite biting analysis in the past, but we've sort of waned from that now. But we looked at mean damaged Varroa over 64-day period during the summer. And you can see here that 3 colonies, 30, 28, and 34, showed a very good mite biting activity, whereas 24 and 6 were poor. And these showed very good survival, long-term survival, and Varroa suppression. The others were not. So what I've done lately is to take a look at doing this visually. It takes a bit of patience, but it does work.
She got away from
So
it right away.
I'll just let you listen to this.
There, that one's got it.
Nate's got it, just chewing it.
I got it.
Yeah.
I got it.
She's chewing it.
So that's an example of where we put a pupa with a Varroa on it, put it in the middle of the bees on a frame, and watch them whether they're aggressive towards it or not. Another thing that we've just started, we've got some unhealthy brood order. Well, it's really a cuticular pheromone, and what we've done is started to do some progeny analysis with that in conjunction with Kara Wagner at University of North Carolina. So this started out late this year, so we didn't get a lot done, but we went through and tested some of our SY26 progeny. So essentially what this is a compound that Kara and her colleagues have synthesized. It's a cuticular component. It's a hydrocarbon, and what they've done is use it to try and test for Varroa resistance, and we've started to do this analysis with ours. In this particular case, we show the control is on the left here with the green spots. or squares, and on the right is 2 hours after spraying this compound on the brood. It's amazing how quick that assay works. So we're hoping that we can use this to really speed up our selection of Varroa tolerant phenotypes. Our assay was 82.22, and Kara used her scan with her computer modeling system software and come up with 84, so we were pretty close. So that's basically some of the work we're doing in the field. Right now, I'd like to turn to some biomarker work that we've done over a number of years. We started with microsatellites, and we actually were able to discriminate between different strains of bees, Russians, New Zealand bees in different areas. Canadian bees show different profiles with 10 microsatellite or 20 microsatellite markers. We've spent more time on microarrays or transcripts on gene expression studies. I've done some proteomics, but I'm not going to touch on that today. But what we find the most informative right now are genome arrays involving signal transduction. So just to talk briefly about our transcriptional work, this was done by one of our graduate students, Sanjay Shang. He worked for several years on this. And what we did here is we identified G4, which is a very susceptible colony, and S88, which is a Varroa tolerant colony, which survived 58 months. And we looked at the transcriptional expression in Varroa infected and uninfected pupa between these two colonies. And you see in this column here is S88 plus, that's Varroa infected over G4 infected. And we looked at the increase in abundance of the transcripts. And 3 of them really stood out: apolipoprotein D like isoform, esterase E4, and cytochrome P450 oxidase. Well, these particular proteins from these transcripts are involved in conferring stress tolerance, particularly apolipoprotein D, which circulates in the hemolymph and is involved in hemolymph clotting mechanisms. It also helps with cuticular formation and so on. Esterases are involved in breaking down certain pesticides and miticides, as well as cytochrome P450 oxidases. So if we ask the question why the survival colonies, why do some colonies survive for extended periods in natural selection apiaries? We've had 2 outstanding ones, S88 and S26. We can say, well, if they're good at grooming or aggressive towards mites, that helps. That's well known. VSH, or hygienic activity, where the bees remove the brood that has Varroa infestations or families of Varroa. Supersedure rates also, I think, help because they break the brood cycle, and our S28 is an example of that. And the stress resistance, I believe, is a big factor in this. And that's what we found out from our molecular analysis, is that these transcripts for these proteins are elevated, and they may play a big role in helping those colonies survive environmental stressors. Some of these survivors are have better foraging activity, better nutrition, and that helps them survive as well. And we're looking into now, which I believe that the ability to cope with viruses is very important in the survival mechanisms. So we'll go to some of the virus work we've done. This was done a few years ago, but we're still working on it. This is an example of an experiment where we look at deformed wing accumulation in some Saskatraz strains with and without mites. So the CT value is on the y-axis to the left, and the higher the CT value, the lower the deformed wing virus numbers. So in this particular case, if we look at SY26, which is one of our longest survivors, we find that in the uninfected, the virus level is very low. If we infect it with a mite, or the pupa is infected with a mite, and we look at head tissue here from the pupa, we see that it's not much different than the uninfected. Same with S23A, and those were both good survivors. If we take a look at susceptible strain, we find that the infected pupa with Varroa have a much higher level of virus. So that indicates that perhaps the ability of these strains to deal with the virus suppression is important for survival. This next slide is the first panel is another measure of deformed wing virus concentration. However, it's a repeat of the last, except at a different time. And we see the tolerant strain again. The infected and uninfected have closer, are not too far apart, whereas the susceptible, again, shows quite a spread in terms of the concentration of virus in the pupa. The next panel is a very interesting experiment, and it's a similar assay, except in this case, we're using an in vivo assay for the effects of miticides on deformed wing virus accumulation in the pupa. The control is with a tolerant strain Y26. We see that there's no significant difference between the treatments. Apivar, Apistan, and Thymovar show similar levels of virus, fairly low in this. If we look at the susceptible colony, after exposure to just fine strips of this in vivo, that the Apistan and Apivar levels are quite high for deformed wing virus, while thymol is less obvious. So what we're right now, we're working on this and we're pursuing this in adult bees within the colonies. Okay, the next thing I want to spend some time on is biomarkers using genome analysis. We started this several years ago and we're quite surprised at the results. So what I'm showing you here is a genome array with each of the spots on the array represents a phosphopeptide. They're 15-mer peptides with the phosphorylation site in the middle. And what we do is we extract the whole bee pupa and probe the array with that. And if the kinases are active for that particular peptide, they light up. And we were quite surprised at how reproducible it was. You see here this set, same the second replicate, third, and we do 9 technical repeats to get our data. But we use, in this particular study, we use infected and uninfected pupa and isolate the whole pupa and probe the array. So after doing the G4 and S88, the 2 extreme phenotypes, we identified 19 phosphoproteins that show differential expression between the 2. And our bioinformatist gathered together the peptides that kinase, or peptides that are associated with innate immunity, metabolism, and stress responses, mainly. And that's where we find some interesting results. This is a principal component analysis of the susceptible and a tolerant. Of course, the S88 was showing the best survivor, and G4, the susceptible. So the tolerant line groups together in one cluster, except for an outlier there that behaves similar to the susceptible. But the important thing to note here is that the infected susceptible, G4, and the uninfected do not group the same. There's a difference in phosphorylation and activation of different pathways. And what we found is that mite infection of the susceptible strain results in suppression of the innate immune system in the bees. So they are more compromised and seem to not survive as long. This panel here is a little complicated, so you got to bear with me here, but this is a genome analysis that we decided to do with colonies showing differential susceptibility to mites. So on the left, we have axis, the y-axis, we have colony survival in months, and that's depicted by the white columns there, colony survival length here. And then the crosshatch red is the mean phoretic mite levels. So we've got those across here showing from low to high infestations. And total honey production over the lifetime of the colony is in the orange cross-hatched areas. And then we have these values called biomarker susceptibility scores. So what we look at here is we see that there's a trend towards, as the biomarker susceptibility score increases, the mite infestation levels increase. Of course, honey production is poor in these because they don't survive very long. But what we hope to do is to be able to predict from the biomarker susceptibility scores where these colonies will fall. So if we have a score that is from our genome that is around tier -2, we would predict that that colony would show some tolerance to mites and good survivability. Now I'm going to leave that and head to our work with OHB on the Saskatraz hybrid project. I know that the reason this started is because we had people, beekeepers in Canada, asking us if they could get some of our stock with that were selected in Canada earlier in the year and at a reasonable price. So what we did was I started this in Orland with David Powell and Ray Oliveris a number of years ago. It's almost 10 years now since we did this. And I think this is important because we can try and get the breeding, the alleles, or what all the selection we've done in the hybrid queens, because the drones carry the genetics of the breeder queen. So we hope to increase the frequency of alleles associated with these traits in commercial populations. We have stock in a lot of places in the world now, and they all seem to be fairly happy with it. And there's some demand or interest from other places too. So this is the team at Road 20. Is Ray and Josh there on the left with Jose Gomez that works with the queen rearing crew, and the ladies there that look after the cell builders and do the grafting. We've had very good results with working with Ray and his team. So just to take a look at the regional or locations, we're doing this sort of stuff. Right now it's going to be -30 in the morning here. I think it's we have a temperature range that is quite extreme and a very short queen production season, whereas Orland, California is about twice, and much better weather for mating queens. Now, briefly, with queen production in Saskatchewan, we focus on production of breeder queens by recurrent selection and close population mating procedures. We have a short queen production season, but it's a good location for selecting for wintering ability and honey production. And we can do up to 2,000 or more queens in the last few years. We're getting better at it every year. So we send about 150 to 200 breeders to be reselected in California. And then in the past, I've gone down and selected 40 to 60 of the best breeders. And those, the team there uses to produce queens on a larger volume, a larger scale. And it's an ideal area for it because if you look at the colonies when they come out of the almonds, they're beautiful. They have high populations, lots of mature drones, and very healthy. So we've been pleased with the results from that. On occasion, the California Tech transfer team and the Be Informed partnership have looked at various breeders, and I'm not going to dwell too much on that except to say I'm glad that they do the freeze kill test, and they come up with some of the hygienic behavior. Most of ours did do pretty well on that. Now I do in the hive stuff, and I go through all the colonies I can. Just to start with, I like to look at the queen. There's something that I see right away here. If I look at the wings and I see 3 nice rings below the ring. I call that the 3 ring rule, which gives me an idea this is likely a pretty decent queen. This is an SY26 and where it's mated at. And I look at the brood, uniformity of the bees, and of course the traits that we've talked about as well. So down in California in about March, I go through the colonies and look at all these behavioral assays. I also like to look at low temperature flight. Queen retinue, I think, gives me an idea of how good the queen is and how well the colony will perform. Pollen storage, propolis, brood pattern, so on. And Varroa assays. And I've started to do Varroa assays within the colony at the same time as I'm selecting. In this case, I'm going to show you what we do. We look at drone brood and see how many drones are in the, or how many Varroa are in the brood, just to get an idea of the infestation in the individual colonies. So I inspect the brood for Varroa, and if I find one, I take and put it on the brood comb and watch how the bees behave. In this particular assay, I look at grooming behavior. That mite jumps on a bee right away. And we'll see here. You can see the bee starting to do her dance and grooming. She's quite aggressively trying to get rid of the Varroa. She eventually does, but it takes her a while. In this particular case, I didn't see any examples of allo grooming, which you sometimes do, where another worker bee or sister will come and help remove the mite. Okay, the retinue is something that I really feel, if you see a poor retinue, that colony is likely going to fail soon. If you see good retinue, it's going to be productive and the queen will survive a longer period of time. It's a reflection of good quality mating.
The bees are.
Just
Look at the retinue building.
This is an example of a queen with retinue building and a laying activity.
She's busy. Oh, there she's laying. Keep on, girl. 1,000 a day. How many seconds did it take her to lay? Oh, she's doing another one right now. Oh, wow. She's laying up a storm, eh? And the bees are right around her.
That sun's out there. We're ready to go.
Look at that. So the bees are really. I don't know if they're encouraging her or what they're doing.
They're just surrounding her though. She's got her head in that cell.
And she's looking at another one, but she laid twice.
She is.
Yeah, she's out now. She's in another one.
Yeah.
By the way. Wow. Look at that. Another
Four.
one.
Okay, I also was invited over to UAE where we'd shipped some breeder queens for to set up a breeding program. And this is a picture of where we had the colonies. It's in the Hatta area, which is near Oman, in the mountains. And you can see they under that cloth we have our breeders are under there, and they keep them shaded because it gets so hot there. That's myself and Sheikh Mana and an Egyptian beekeeper that was working with our colonies. I went through a number of colonies there, but I was impressed by this particular strain. This is a Apis mellifera yemenitica, and it was a very gentle hive because some of the bees over there are not gentle. I was stung a few times looking at bees in different areas, but the brood pattern was nice in this particular colony. And you see the queen there, and there's a drone, a Saskatraz drone, over visiting Mrs. Yemenitica, and you see their drones are quite a bit smaller. These bees are not that great for productivity, but they're certainly heat tolerant and good survivors in that climate. Okay, I'm going to switch over now to it's getting near the end here, but I want to look at miticide treatments in stock selected for Varroa tolerance, because I believe that if we select for survivor colonies, which show excellent tolerance to Varroa under our conditions, but we have a difficult time stabilizing that trait in the progeny. And once we get into the bees are mixed with other populations, they soon lose their tolerance. So commercial production still requires miticide treatment protocols. So and we find that we can improve the efficacy of these treatments if we use stock bred for tolerance. So I'm going to show you an example of an experiment we did some time ago, and we're redoing this. But these colonies were in 3 different apiaries, and we measured the mice per 100 bees, you see on the y-axis. And these are 3 populations, Saskatraz and 2 unselected populations. And before treatment, this was in the fall, we treated them, and we didn't wait long enough, but there was some reduction in mite levels. But the dramatic change is in the spring. In the spring, the mite levels in the Saskatraz colonies were very low, whereas in the others, they were significantly higher. But the important thing was the survival. The percent survival of the selected colonies was 85%. That of the unselected was much lower, and likely most of the mite infected colonies here died. So that's just an example of the difference. So now we've been experimenting with combined mite treatments for a number of years now, but I'm just going to show you quickly what 4 different treatments. So if you look at the left axis, that's phoretic mites, the y-axis, and the blue bars are the initial concentration or the initial levels of Varroa in these colonies to be treated with the different combined treatments. You see they're not significantly different. And if we look at the first year of treatment, the spring of 2016, this was done. In the fall, Varroa levels are very low. So all four treatments worked well the first year. The next year, we find that they work well too, but in the apivar, we're starting to see more increase in mites. But I'll just skip over to the final or the take-home message here is that in 2018, we see apivar treatment alone that the mite levels are staying a little higher than we'd like, whereas in the apivar plus oxalic, they're much lower. In the apivar and thymol, they're not detectable, or in the apivar plus thymol plus oxalic treatment combined. But the other important thing here is that after this series of experiments over 34 months, we see that we still have decent survival in the combined treatment regimes, but apivar we're down to 27% of what we started with. So that's something to think about. This is the last slide here that I want to deal with today is about a longer term analysis. And again, we're looking at mites infestation on the y-axis. And in the top panel, we look at apivar plus oxalic. I'm just going to concentrate on the last 4 years here, 2017 to 2020. We see that spring treatment, we use apivar and oxalic because thymol tends to reduce brood production in the spring. But we see very low levels with this treatment and consistently. In 2017, we had 80% or more survival. The same in 2018, 2019, 2010, we had, or 2020, we had about a 93% survival rate. In the fall, we use apivar and thymol, and we see the same thing. Very low levels of infestation, and survival rates are good. But this is all with stock, either Saskatraz hybrids or with our stock. So to summarize what we've gone over here, we can select families or strains with good honey production, wintering, and Varroa tolerance, but it's difficult to balance these phenotypes. Varroa tolerance is variable in the progeny because of the nature of bee genetics, and unless everybody has tolerant bees, they're going to quickly lose that tolerance because I believe the VSH phenotype requires it in homozygous state to give the best expression. Our focus is aimed at stabilizing mite tolerance in our work in progress, and we're optimistic we can improve this by using extensive progeny analysis with marker-assisted selection, particularly the Genome assay and the transcriptional assays, and UBO assay. I'm hoping will speed up our selection process to identify Varroa tolerant daughters. We're also working with Dr. Mike Simone Finstrom and the USDA people in Baton Rouge, and we have they've already screened some of our strains for virus susceptibility, and that should shortly be published. And what we're working on at the moment here too is studying the effects of miticide treatments on colony virus infections and what impact that might have on honeybee health and Varroa population growth. I'm a little suspicious that we're hurting our bees with some of these treatments, and they become more susceptible to regrowth of the Varroa population. So I think that's about it. So we worked Meadow Ridge. We fund our breeding program by selling our bee stock, and we've been working with Saskatchewan beekeepers for a long time, since 2004. We have some funding now from APIS-M for the virus work with miticides, and we're working with USDA. Vito has worked well with us for many years. Philip Griebel and Scott Napper, Wayne Connor. Toxicology I didn't talk about. We worked with Dr. John Giese over several years for that, and other people that were involved with the program are listed below. So I think I'll stop there, and if you have any questions, I'd try and answer.
All right. Well, thank you
Thanks.
very, very much, Albert. That was a great talk and right on the money in terms of time. So I applaud you. Medhat's going to pull a couple, there's 4 questions on Q&A. Medhat, 2 issues. Panelists can't ask questions for Q&A. So Melanie from Zia Queens had some questions. So why don't we start, Medhat, with you asking some of the questions from Q&A. Then I'm going to ask Melanie for questions, and you can go take a quick look at chat to see if you got some questions there that you can toss to Albert. So, and remember, we also have Ray Olivares here on the panel if there's something comes up that might apply to his part of this project. So go ahead, Medhat. Medhat? You need to turn your microphone on. There you go.
Yep, it is on. Yeah.
Okay.
Thank you, Albert, for the presentation, and it's a great deal of work gone into Saskatraz, and thanks for sharing all of this work with us. I have one question here. What are the long-term traits that you are trying to establish in Saskatraz breeding program?
Long-term traits? Well,
Yeah.
honey production, wintering ability,
Yeah.
Varroa tolerance, and resistance to root diseases are the main ones. Virus, we're just starting, but we've seen some promising results in some of the long-term survivors. Are
these traits designed for certain geographic regions?
You know, actually, honey production seems to work well wherever we've sent our stock.
Right.
I was surprised in Ukraine, they're really pleased with how the honey production is associated with our stock, and in the hybrids that they produce, they've been showing quite substantial increases in their production. Also in Afghanistan, they're quite pleased with them. Turkey, Istanbul, we shipped some to years back. Lebanon, I don't know about too much. UAE, Iran has had good success, and honey is valuable in those countries.
Right.
So they like that. So that's one trait that seems to have some stability.
Have you got some estimate about how much increase in the honey production in some of these countries? Like 5%, 20%?
No more than that.
Oh,
actually,
okay.
Ukraine 40 to 50%, they say.
Okay,
I don't
great.
know. You know, I'm just quoting what they're saying. I don't know for sure, but I have people that I work with over there that I could ask that from.
Okay, the second question here, it says, does hive size matter on how long the queen might survive and reproduce? For example, feral colonies, usually they are smaller than the managed colonies, and they can usually live longer. So do you think there's a correlation between the hive size and the queen productivity and longevity in other ways?
I know that some hives that are smaller can cope better with Varroa. In terms of the work that we've been doing, we put lots of selection pressure on honey production. And you've seen the S28 there is still producing good honey, and that hive has been there since 2012. And their population increases dramatically in the spring, but they go down to a reasonable size in the fall. So one of the things that you want to have in your breeding stock is where a colony prepares itself by winter by reducing the population down to a size that they can survive with the storages that they have. Then you need that spring population to build fast. So, you know, that's what I think.
Yeah. I guess the question comes from when you show some hives, like the ones where in your slides, like 4, 6 high, and and in lots of places, they usually 2-3 high. So the question, how long a queen in Saskatchewan will survive under this kind of high production rate comparing to other places?
I've seen some queens survive 3 to 4 years
Right.
marked. Unfortunately, the marks we put on are often removed pretty quickly by the bees because of their hygienic behavior or grooming.
Yeah.
But we've had some where the wings were clipped, and I've seen them survive up to 4 years, but I think that's unusual. Usually they supersede at some time or another during the time, but.
Actually, this brings me to a question. It was in the chat early on from Melanie. I think in your presentation, you mentioned some queens lasted 58 to 70 months.
Yeah, so not queens, colonies.
Colonies.
Yeah.
So, okay. Are these, so the queens might be changed or something replaced?
Oh yes. Yeah, that's an important thing. Like, for example, S28 that I talked about in selected for honey production.
Mhm.
We see her coexist with a daughter quite often. I've seen that since 2006. That trait is still there. And so supersedure like that and maintain, it's kind of a Russian trait. They used to maintain a lot of cells and emergency cells. And in this particular case, this colony coexists with a daughter.
Yeah.
So that's good protection against queen failure. So I've seen that on more than one occasion. So that helps with maintaining the colony is the important thing. The queens I've seen last for quite some time though, up to 4 years with clipped wings, so I know who it is. Often the marks are gone after a few years, but
This is
does
our
that
kind of
answer
person.
your question?
I think so. Like the question right now, like, have you looked at your queen's number of sperms in the spermatheca? Like,
I haven't lately, no. I'll
Yeah.
let you do that, Medhat, next year.
well, I'm saying that because we see lots of variations and definitely it's quite interesting project. Yeah.
What I feel is that our best mating is in August.
Yeah.
Late July, August, because we have more mature drones then.
Yeah.
And higher populations. We have a difficult time having enough drones in May or
That's
late
correct.
May even. And then your mating you would expect to be poor.
Yeah.
So the later in the season in our country, the better mating because of the population of drones. Like it's beautiful to see that in California, those colonies coming out of the almonds are just booming with lots of drones, healthy drones.
Yeah. Do you expect your girls will do better in Southern Arizona?
Or what? Sorry, I missed
Your
that.
girls, your queens.
Oh, my girls. Oh,
Yeah.
I don't know. They did okay over in Iran and in UAE. At least they survived the heat, but they're not as heat tolerant as their stock. So I guess we have to take some there to see.
Okay. And then yeah. Was your research about miticides and your Saskatraz queens and bees, have you come to a recommendation what is the best combination of miticides can be used effectively in Saskatraz bees?
Yes, our data shows that the combination of Apivar 2 strips, 1 of thymol works the best.
At what time of the year? Like that's
in the fall.
In the fall.
Yeah, we feel, well, thymol works in the spring too, but the thing is thymol, if it does inhibit brood production a bit early on.
Yeah.
You know, it does slow that down a bit, but it works fine. We're trying to produce some of our own thiamol pads for treatment, just to in combination with Apivar, and to look more into this. But like, we're just using 2 strips, and I think the more Apivar you use, maybe the harder you on the bees. And you know, there's concern about some Amitraz resistance or Apivar resistance, but if Varroa was resistant to Amitraz, the bees, it would be terrible. I mean, they would just take over, and they haven't done that. So we see these sporadic cases where Apivar doesn't work. One of the reasons it might not work is because the bees are not able to synergistically work with the treatment to reduce the Varroa levels. You know, from my experience in a previous life, anything that become resistant, it was all over. Well, look at the with Apistan. Once Varroa become resistant, it doesn't work at all. Same with Checkmate. So I think it's more complicated here in the I'd like to see what's happening in the hive with these miticide treatments. I have a hunch that we're hurting our bees, and we're decreasing their ability to deal with viruses, and that's making the bees sick, and that makes them in turn less able to cope with viruses, diseases, brood diseases, etc.
Gentlemen, I think we're at the top of the hour.
Yeah.
I know there are questions.
I have a question here, Jerry, from Randy. Quickly, I'd like
Yes.
to squeeze it in.
Sure.
Regarding hurtability, what kind of percentages of fully resistant daughters colonies can be seen from fully resistant mothers? Like, how is that hurtability moved from the mother to the daughter? How much percentage of hurtability you can see?
That's a good question. We're working on that, and I'm disappointed in the heritability of Varroa tolerance. The reason being is I think that VSH type phenotypes with has a big influence are likely a lot of cases in the heterozygous state. They need to be more homozygous. So our plans with our progeny analysis is to try and select colonies which have very high VSH phenotypes, so that we can cross those to the matrilines that also have very high Varroa tolerance, so we get the right combination and frequency of alleles in those populations, so we can improve the resistance. So maybe in a couple of years they'll be able to answer that better, but right now it's not great.
Well, I thank you very much. I would like to, Ray Oliveris, do you have any final comment you'd like to make? I mean, Arnold, Albert's been given the great briefing, but you are his partner in California. Do you have a final comment?
Yeah, I just keep learning every time I listen to Albert. You know, the one thing, I just have one comment that I've noticed in our own bees, and just when you talk about just that single selection for honey productivity, if you think just on the common sense level, if you have bees that produce a lot of honey, they've got to produce a lot of bees to produce that honey. That means they got to be good pollen gatherers and nectar gatherers to grow those bees. And it's a cause and effect. And when those bees are that healthy and able to go out and get pollen and nectar, it just snowballs, and it just builds in forgiveness. And I believe Albert's definitely onto something with these, the weakened immune systems due to whether it's fungicides or miticides, is environmental stressors. And because I see the same thing, I hear about resistance to Amitraz, and but it's not consistent. and it's spotty, and there's all these variables. And I think a lot of it has to do with the bees being in a weakened state at the wrong time, wrong place and time. And, you know, it's just going to take years to understand that.
Thank you, Ray.
Okay.
So we really need to move on to the general meeting because we've got to do that to meet our requirements of our nonprofit. And also we have to have some elections. So Albert, you and Ray, some of the panelists couldn't ask their questions through Q&A, so they asked them on the chat. I suggest, if you don't mind, because we're going to stay alive here and go right into move along to our general meeting. If you have the time, if you're not completely worn out for the day and so on, take a look at the chats and the chat and questions, and maybe you can answer a few more that we didn't get to. So I really thank you, Albert and Ray. I think that was a really nice capstone talk for the year, and I'm thrilled that you were with us today. And we had about 130 people at attending this one. So in the middle of the holiday season, where people got Christmas parties and so on, that's a pretty good number. And I'm sure lots of them will be going to the recordings at their convenience. So Albert, I thank you. Ray, I thank you. And Jaylene, do you want to address the poll? And while Jaylene's addressing the poll, Rich Morris, you can wake up your system. And as soon as Jaylene talks about a poll we have here for everybody, you're on. So Jaylene, you want to tell them about the poll?
Sure. We just have a quick poll that we'd like people to fill out. This is a pre-election poll. It's just a little survey if you would fill it out, and I'll share the results once people are done. And let's hear from Rich.
Well, thank you. Thanks, Jerry, for inviting me to give you money to help sponsor this.
I see, though, you're wearing one of your colorful shirts. So,
well, you know, it's my thing. So, yeah, no, I've been keeping bees about 15 years, and I just constantly realize that the more I learn, the less I know. It's just fascinating, and it's been a really great journey for us. We started about 6 years ago. We just passed our 6th anniversary, actually, and you know, during that time, we've met a lot of the people I see on the panel here, and a lot of you out there, and it's been fabulous. I've really missed going to the meetings the last year, although, you know, it's certainly a lot less mileage. But what we have going on at Broodminder, you know, there's a team of about 6 to 8 of us that the internet-based company we've met around the internet and whatnot. And we just, you know, keep working on things and trying to make them better. You know, we're making still the temperature and humidity sensors and weight sensors. The real tricky part has been, you know, this is a very simple data logger, but when you're in a beekeeping system, you know, things are moving all the time, and you have different data coming in from different places. So it's been a real challenge in that way, but we've so far shipped about 22,000 devices around the world, and we're now at the point where it's getting to be a lot of fun. We're getting a couple of more people on the team. Recently, Anya McGurk, a data scientist in North Carolina, retired, and so we're just starting to pour through this big mass of data. You know, we got hundreds of millions of data points now. And one of the more interesting things I think we talked about at the meeting a year ago was that, you know, we still feel that internal temperature is the most telling and most cost-effective measurement of the hive. And watching that, we switched from watching every hour or every 15 minutes. We're now looking every minute and watching for those changes. And in particular, the one that's most interesting is when they're under thermal regulation, you know, things happen. And so in particular swarms, we see, you know, about a 4-degree quick rise in temperature. We believe their thermal regulation system is basically just gone out of kilter. We talked with Jerry quite a bit about this. He because he has so much experience in it. But when we detect that, we know something dramatic happened. And then, we pass that through our system and, generate text messages and those sorts of things. But we're now going through those more with a fine-tooth comb and looking for those signatures and seeing. We see that, swarming, queen mating flights show up, certainly other events, mite treatments, we see these sorts of things. So it's getting fun. You can see the shelves behind me are sort of bare. We've been shipping a lot lately and keeping up with it, but around Christmas we have a lot of shipments, and I just want to encourage organizations, especially like WAS. I think it's having a bit of a resurgence here, and I hope it continues, and we're glad to support it. Thanks a lot for inviting me. And good luck.
Yeah, thanks, Rich. And there's a couple pressing questions. First person wants to know where they can buy your shirt.
Well, and this isn't, I've got some beekeeping ones I forgot to put on this morning. I've actually, Facebook targets me now for Beehive/Hawaiian shirts.
And then the other one was they want to know what kind of honey and nectar that shirt produces.
Only the best.
Only the best. Well, thank you, Rich, thank you for being here tonight and giving a little briefing on broodminder. I know at least one of the panelists on here has been up in the Yukon territories. Etienne has been using your broodminder and doing some really
interesting. Is a master of our equipment and a master of beehives.
And he's got a great test facility up there in the Yukon territories.
yeah, we're very close.
All right. So
okay,
we're going to move on.